Our next speaker is Don Kufe from Dana-Farber, who's been a longtime leader of clinical pharmacology and is going to talk about MUC-1 biology and target for therapy.
Thank you, Dan. So I'll be talking about MUC-1, an oncogenic protein that is not a receptor tyrosine kinase and has been thought for many, many years to be an undruggable target. So, I'll be talking about the scientific background that supports MUC-1 as a potentially attractive cancer target, and our development of agents that block MUC-1 function. So the early work on MUC-1 was focused on its overexpression in about 90% of human breast cancers. And that led to its characterization as a high molecular weight glycoprotein, and led to the development of the CA15-3 assay for its detection in plasma. And then with the cloning of the MUC-1 gene, we found that it contains a unique structure of variable numbers of 20 amino acid tandem repeats that are extensively glycosylated. And that structure has now been found in a family of some 20 proteins that are known as the mucins. And among others, CA125 or MUC-16 is a member of this family and overexpressed in ovarian cancer. Now, the mucins play a very critical role in protecting our epithelium, which is a single cell layer that's exposed to the external environment. For example, lining the respiratory tract and the gastrointestinal tract. Our epithelium also lines the lumens of ducts in specialized organs such as liver, pancreas, and kidney. And as a result, this epithelial layer needs a very robust defense mechanism for its protection. So the mucins come in two different forms, secreted and transmembrane. And the secreted mucins form the mucus gel, a physical barrier that represents the first line of defense. And the transmembrane mucins contribute to that barrier, shown here, but also, and importantly, signal stress to the interior of the epithelial cell, promoting growth, survival, and repair of the epithelial layer. Now, MUC-1 is a transmembrane mucin. It's synthesized as a single polypeptide, undergoes autocleavage in the ER, and then moves to the cell membrane as a heterodimer. The N-terminal subunit, MUC-1N, contains these tandem repeats that are extensively glycosylated. This is the mucin component of the heterodimer. And it's tethered to the cell surface in a complex with the transmembrane subunit, or the carboxy-terminal subunit, MUC-1C. Now, early attempts at targeting MUC-1 were focused on the N-terminal subunit, because it's easy to make antibodies against this subunit. And these attempts at targeting MUC-1 were uniformly unsuccessful, largely because this subunit is shed from the cell surface. Now, more recent work has focused on the transmembrane subunit, which signals stress to the interior of the cell and induces transformation. Now, it's also important to point out the positioning of MUC-1. And for example, here, our epithelial cells, of course, are polarized with the apical border facing the environment and the basal lateral borders expressing receptor tyrosine kinases. And MUC-1 is positioned at the apical border, affording protection against the external environment. And with stress or with transformation, there's a loss of polarity, and now MUC-1 is expressed on the entire cell surface and can form complexes with RTKs. Okay, now give you an example, this is a breast tumor that is stained with an antibody against MUC-1. And you can see that in the center is a normal duct, and you can, there's a fine rim of staining with this antibody against MUC-1 along that apical border of this normal ductal epithelium. Now, in the foci of carcinoma cells that surround the normal duct, there's more intense staining. MUC-1 is expressed on the entire cell surface and accumulates in the cytoplasm and is upregulated about 50-fold in this case, which is fairly consistent with many breast cancers. Now, MUC-1 over the years has been found to be expressed in other types of tumors as well. For example, non-small cell lung cancer, castrate-resistant prostate cancer, gastrointestinal tumors, particularly pancreas, and gynecologic tumors, for example, ovarian. And there's a huge literature on MUC-1, and its overexpression in general is associated with more aggressive disease and metastases. And MUC-1-induced gene signatures are associated in breast, lung, and prostate with poor, disease-free, and overall survival. Now, somewhat paradoxically for an epithelial antigen, MUC-1 is also found in hematologic malignancies. Virtually all primary multiple myeloma cells express MUC-1, as do the cell lines. And certain lymphomas, for example, anaplastic large cell lymphoma. In CML, MUC-1 is detectable in blast, but not chronic phase cells, suggesting that it may be associated with more aggressive disease. And David Avigan at the Beth Israel Deaconess Hospital has recently shown that MUC-1 is expressed in the myeloid leukemic stem cell, but it's not detectable in normal hematopoietic stem cells. So taken together then, this expression of MUC-1 in very diverse carcinomas and certain hematologic malignancies has made it a potentially attractive cancer target. In addition, the MUC-1 knockout mouse has no phenotype, indicating that you might be able to target this molecule without adverse effects. And importantly, the MUC-1C subunit is an oncoprotein. And again, just to remind you that the MUC-1N subunit is the mucin component. That's out of the story here, and we're focused on the transmembrane subunit. Okay. Now, the evidence for its oncogenic function is just listed here. If you overexpress this subunit and indeed the cytoplasmic tail, it's sufficient to induce anchorage-independent growth and tumorigenicity. There are transgenic models, for example, MMTV MUC-1 transgenic mice generate mammary tumors of all sorts of diverse histopathologies, and some of these tumors are metastatic to the lung. And overexpression of MUC-1 also blocks death in response to stress, for example, in response to DNA damage, reactive oxygen species, hypoxia, just to give a few examples. Perhaps recapitulating its normal function to protect our epithelium against environmental stress, such as toxins, microorganisms, even low pH in the stomach. Okay, so with that as background then, how do we target this guy? So shown schematically here is kind of a summary of what happens to this subunit in the cancer cell. So here it is positioned at the cell membrane, forms complexes with RTKs that are mediated by a galectin-3 bridge. And as a result, the cytoplasmic domain of this subunit gets phosphorylated. It moves into the cytoplasm where it forms dimers. And the dimers are necessary for its import into the nucleus, where it interacts with transcription factors and activates genes involved in growth and survival. And the dimers are also necessary for transport of MUC-1C by HSP70 and 90 complexes to the mitochondrial outer membrane where it protects against activation of the intrinsic apoptotic pathway. So we reasoned that the Achilles heel of this subunit might be its dimerization. And in order to identify amino acids responsible for that function, we needed to turn to the structure of this subunit. And it's shown here schematically. This is the piece outside. It's 58 amino acids, has a 28 amino acid transmembrane domain, and a 72 amino acid cytoplasmic domain. Now, this is the amino acid sequence of the cytoplasmic domain. It's phosphorylated by numerous kinases, just a few of which are shown here. For example, MET phosphorylates tyrosine 20, which is a YHPM site that's a consensus when phosphorylated for binding of PI3K SH2 domains, leading to activation of the AKT-mTOR pathway. EGFR and SARC phosphorylate tyrosine 46. ABL phosphorylates tyrosine 60. The Wnt pathway kinase GSK3 beta phosphorylates this serine. And there's a consensus sequence here for binding of the Wnt pathway effector beta-catenin, allowing this subunit to form a complex with beta-catenin and TCF4 to drive cyclin D1. Now, mapping the sequences, the amino acids for the dimerization led to the finding that these two cysteines here in the cysteine-glutamine-cysteine motif was essential for dimerization. So we reasoned that if we could make drugs against this motif, that we might be able to block MUC-1 function. And the first approach we used was a cell-penetrating peptide drug. And so, for example, you can link transduction domains to peptide sequences. And so our idea was that we made this peptide drug, penetrated cells, binds to the cytoplasmic domain of the MUC1C receptor subunit, and blocks its ability to form a dimer. It's a pretty simple approach. Okay. And then one of those drugs, GO201, I'll tell you a little bit about, another one, binds this region, actually blocks dimerization, and shuts off these two pathways as a result. So let me just tell you a little bit about what we did, the evolution of these peptide drugs. And personally, I think that there are going to be more and more peptide drugs entering the clinic to target previously undruggable proteins. So, for example, here, 201 was the first one we made. It was L-amino acids and incorporated the first 15 in the cytoplasmic domain. And the control for GO201 was this peptide here where we mutated the two cysteines, so to alanine, so it's AQA. Some of the work with 201 has already been published, so I'll tell you a little bit more about GO203, where we moved to D-amino acids. Still has the transduction domain. It's shorter, and the D-amino acids afford more stability and are less likely to develop inhibiting antibodies. And the control for GO203 is this peptide here, again, with the mutating the critical cysteines. Now, just to show you a little data with 203, this is a study that we're doing with Maurizio Scaltriti and Jose Bazelga, using their breast cancer cells that overexpress ERBB2, SKBR3, for example, and treating these cells with GO203 peptide at 5 micromolar, associated with an initial inhibition of proliferation and then loss of viability. And this pattern is similar in the SKBR3 cells that are resistant to Herceptin. And we have similar results in the BT474 cells that are sensitive and resistant to Herceptin. So now we're interested, of course, in what the mechanism is here. And in contrast to Herceptin, which has little effect on the phosphorylation of ERB2, treatment of the SKBR3 cells with GO203 associated with downregulation of phospho-ERB2 without an effect on ERB2 levels. And that response is similar in both the sensitive and Herceptin-resistant cells. You can see here a downregulation in phospho-ERB2. And we also see a downregulation in AKT in the sensitive cells and in the resistant cells. And as we've heard, it's important to block both pathways, AKT and ERK. And you can see that using this approach, we also see downregulation of ERK activation here in the sensitive and resistant cells. Now, if we take 203 into animal models, this is a breast cancer, estrogen alpha expressing breast cancer xenograft growing in nude mice. These are established tumors here, about 100 millimeters cubed. We're treating every day for 21 days. This is a vehicle PBS. If we treat with 203 at 10 milligrams per kilo, there's no effect. If we go 20 milligrams per kilo, you see there's inhibition of growth during the treatment period, and then the tumors regrow. But if we go to 203 at 30 milligrams per kilo, there's inhibition of growth during the treatment period, and then these tumors slowly regress over time, and they're long-term survivors. Now, this type of response is not restricted to breast cancer. And for example, here's H1975 non-small lung cancer cells, which, as you know, are EGFR mutant, resistant to TKIs. Here again, xenograft model, established tumors, treated with PBS, treated with the control peptide CP2, which does not block MUC-1, no effect. And here treatment with 203 at 30 milligrams per kilo, a marked inhibition of growth, and then long-term survivors. And as we saw in the breast cancer cells, you can see the treatment with 203 is associated with inhibition of AKT and mTOR, indicating that part of this effect may be mediated by suppression of this pathway. Okay, so based on these and other findings, we took 203 to the clinic, and the clinical formulations known as GO203-2C. The phase 1 trial is underway. We've treated 8 patients, no toxicity so far, no anaphylactoid reactions, and the trial is ongoing. As backups, we've made alpha helical peptide drugs based on the sequence of GO203, and we've identified small molecules. We set up a dimerization screen and went to the HMS facility, screened about 5,000 compounds, got a number of hits, several of which are natural products, interestingly, and they block dimerization. And then we've moved those hits now into a structure-based lead design, making additional small molecules. So these are backup compounds. Now, one can target molecules in the cell membrane, both in the intracellular domain and potentially in the extracellular domain. So we reasoned that perhaps we could make drugs against the 58 amino acids that extend outside the cell. And here's the amino acid sequence of the 58 amino acid extracellular domain. Here's asparagine 36 that binds to galectin-3 that links this subunit to various RTKs. And so two approaches are being used, of course, by pharma these days. One is to make antibodies against the extracellular domain, and we've done that. against critical regions. The other approach is to make a soluble receptor, okay, like a VEGF trap, where you take the extracellular domain, you link it to an FC molecule, and we've done that, and I just want to show you a little data there. So the soluble, we call it the soluble receptor. I guess you could call it a MUC-1 trap if you wanted. But it has dose-dependent effects in vitro, and it's active in xenograft models. And I'll just give you one example. So here, these are ZR breast cancer xenografts treated with PBS here, and a dose of 1 milligram per kilo of this soluble MUC-1 receptor has no effect. And if you go to 10 milligrams per kilo twice a week for 3 weeks, you can see a slowing of tumor growth. And then if you increase the dose further 3 times a week for 3 weeks, you get more complete inhibition of growth, and here we're getting some long-term survivors. So there's a dose-dependent response of this agent in vitro and in vivo. Okay, so to summarize then, we think that this transmembrane subunit of MUC-1, again, not the mucin component, is druggable in the extracellular domain with soluble receptors, and we have now a panel of monoclonal antibodies that react with this region, and in the intracellular domain using peptide drugs and eventually small molecules. So we think that targeting this MUC1C subunit may be broadly applicable to diverse carcinomas and hematologic malignancies, in that overexpression of this subunit, is a common alteration in many cancers. And I hope I've convinced you that it's definitely a druggable target in using agents against the extracellular domain and the intracellular domain. So I'll stop there and answer any questions. Thank you.
Excellent. Questions? Let me start with one question. Is there any insight about biomarkers that would identify subsets that are sensitive to MUC inhibition?
There are a number of antibodies that we've made now against the cytoplasmic domain. So we can stain tissues with those biomarkers there and hopefully predict which patients will respond. But you can have high levels of MUC1 expression, and those tumors would be responsive to these inhibitors. Could also have low levels, and they can be responsive. So it's not necessarily dependent upon amount that's expressed. Excuse me.
Don, the peptides have to get into the cells to work.
Yeah.
What can you tell us about, first, the pharmacokinetics of these small peptides, but then secondly, their ability to get into cells?
Yeah, good question. So these peptides have short half-lives because they're cleared by the kidneys. And so you have to give them frequently. And we can deliver them subcutaneously. The clinical trial's IV. That was sort of a proof of concept. But we've done animal models now delivering sub-Q. So that's one way to get around this. The other way to get around it is to put them in nanoparticles, and that's a terrific approach. You give it once a week and they're effective. So I think we've solved that problem. To get the peptide into cells, you need that transduction domain, and you need to have 1 micromolar levels. It's like you have to push it in, threshold. And whereas if you use the alpha helical peptides, a la Greg Verdine and Lauren Walensky's, you don't need the transduction domain. The alpha helical peptides will go right in. So you can get rid of that component of the drug. And they're very stable. And it's possible you could give these alpha helical peptides orally. The problem is the solubility. You lose solubility with the alpha helical peptides.
The nanoparticle would be the solution to that.
Yeah. The nanoparticle approach really, really interesting for these peptide drugs. As I mentioned earlier, I think the peptides will be, we'll see more and more of them in the clinic for undruggable targets. Okay?
Don, I was
Yeah.
just wondering if in your phase 1, are you following the T-cell responses also from the perspective of, I know that this is targeted, but also are you activating another arm in this setting?
We're not. We're looking for antibodies. You know, these deep peptides, they don't present by MHC complexes, so, or very little. I'm not sure you'd get a T-cell response, and general experience is that you don't get antibody responses against them.
This can be briefly described as, do you have any systems where the drug doesn't work? And in more detail, do you know, does it have to be binding to a receptor in order for the drug to work? And particularly in ZR75-1, what do you think MUC is doing? What are you inhibiting when you give your dimerization inhibitor?
Yeah, this approach does not work in, so if you take prostate cancer, for example, hormone-dependent prostate cancers are not dependent on MUC-1. Those cancers are not sensitive to this peptide. But as the tumors become castrate resistant, that's when you start to see the sensitivity to these agents. The mechanism, I think, is in part related to these complexes with RTKs at the cell surface, but not totally. You can block MUC-1 function at the mitochondrial outer membrane and in the nucleus. So it's not all RTKs, and I don't think it's all inhibition of PI3, AKT, or ERK.
Thanks very much,
Yeah.
Don.